Hello, thanks a lot for watching this video. In this video, I will give a short description of our work entitled Predicting Microbial Composition through Deep Learning. The motivation of our work is that rationality managing the microbial community requires improving our ability to predict how different species assembly affect the final species composition of the community. However, making such prediction remains challenging because of our limited knowledge of the diverse physical, biochemical, and ecological processes governing microbial dynamics. So in this work, we developed a deep learning framework that automatically learns the map between species collection and community compositions. So mathematically, we will solve such a problem given the species assembly, which is a binary vector, where ith entering the ij equal to 1 if ith species is present. Otherwise, they are equal to 0. Meanwhile, we have the species collection, which is also a vector, where pi is the relative abundance of the ith species. Then we will find a map between the species collection and the species composition. So we developed a deep learning framework based on neural ODE to approximate such map. Given a bunch of training samples, CNODE is trained to predict the species composition based on species collection, and the parameters of the CNODE are adjusted according to this loss function, which is defined as the average dissimilarity between predicted composition and true compositions for all of the training samples. To use the CNODE, we need to prepare two files for the training set. One is a matrix Z, where entry Zij equal to 1 if species I is present in the sample J. Another one is a matrix P, where Pij is the relative or absolute abundance of species I in the sample J. To test the CNODE, we need to prepare the same two files for the test set. So here is an example input files for the CNODE. The left is the species collection matrix Z, and the right panel is the species composition matrix P. Our CNODE code can be downloaded from this GitHub link. To run the CNODE, users also need to install the Julian modules, and the version of each module can also be found at our GitHub page. Then users will also need to specify the input file path and revise the parameters if needed, such as the epoch, batch size, and the learning rate of the CNODE. CNODE will output 5 types of the files. W is the weight matrix of the neural ODE, and PTRN is the normalized composition matrix of the training set. ZTRN is the normalized collection matrix of the training set. QTRN is the predicted composition matrix of the training set, and LTRN is the loss of the training set. Thanks again for watching this video.